So I think it's time to get started into data visualization principles for enhancing effective scientific communication. This is a long title. We will keep it shorted as data visualization principles for now. Just so you know, everybody knows my email is here for reference. I work at EMBL, but that will be unfortunately leaving in October. So I leave my second long-term email down in case somebody has questions or discussions down the road. All right, so as I said, short title, let's get started. When I started thinking about this webinar and how visually I wanted to approach it, I started playing with this color wheel and, adding some kind of fun lines and dots. And, I just wanted to make something fun related to the topic. And then I thought, well, this is great for a cover slide, but how do I use this throughout my presentation? I wanted to use this as a timeline of what type of topics we are going to cover. So I thought, well, let me simplify it a little bit. We're going to talk about 5 different sections. So let's simplify it into 5 different color areas. I thought, well, maybe this is a bit too complicated. Let's simplify it even more. And then I make the final decision of let's fully simplify it. So if there is something at the end of this webinar that I would love for you to remember is to keep things simple. It's really easy to complicate and add layers of complexity in scientific visualizations, but if you keep it simple, things will probably work out better. So keeping this simple visual graph, we will be talking throughout this hour about 5 different topics, or we will have 5 sections. Visual thinking, we will for sure cover color. If everybody knows, for whoever knows me, this has to be here, is my favorite topic. We will be talking about graphs in a general way, but hopefully you can apply to your area. We will hopefully have a little bit of time for practice. I wish this could be interactive, but hopefully it's even useful if I do it with you listening. And then we will have a final section of Q&A. So let's get started in visual thinking. The first question that we could start with will be why we care about this, why we visualize science. There are 2 answers. There are probably more, but I summarize it in 2 different answers. First, to explore the data while the scientific story is ongoing. So you are in the lab, you are collecting information. You want to explore what are you collecting. You probably need to visualize data to understand it. The second is to explain, once you have collected your data, the results. Of course, audience will play a big role here. There are different ways to approach this, but no matter what, you have to visually translate the science. If we think about different topics and different areas, and this is going to be really quick, but just a run through of why visualizing science matter. Why in surgical visualization, we don't just use pictures. And this is an example, hopefully it's not too sensitive for everyone, but it's a surgery of the cervical vertebra 1 and 2, and a visualization on the side. When you are showing pictures, you see a lot of blood, you see a lot of fluids happening. If you get other type of visualizations like X-ray, expert for sure will understand what is ongoing. But if you have to explain this to other type of audiences, you might want to think about something else than pictures. Another example, this is a cross-section of a female pelvis. And this is an example in anatomical visualization, why we care about visualizing science. Why, again, we don't just use pictures. Well, it might be a bit difficult to understand what type of organs are going on, and that's why we use illustration and we use color in this example. It was just, it's a paper published a few years ago, but it just separates different organs within a cross-section of a female pelvis to understand what we are talking about. For the same reason, I could ask you why we don't just use numbers. What we are discussing today, there's nothing new about this. This is a paper from 1973 that already start thinking of scientific visualization, data visualization, and why we care. If we see these different 4 groups, we probably can see a trend. If we take our time, we can see what is happening in each section. But if I show you a plot of each of the groups, you right away can get the message. And this is why we care. This is why we are trying to improve daily on how we portray data to different audiences and why this matters. So that said, there is a very, very important concept within data visualization that we need to clarify, and it's the concept of salience. So what is salience? First of all, let's look at the definition. Salience is a visual quality that sets an object apart from its surroundings. How we translate this? Well, let's look at this picture. If I show you this and I ask you how many women you have in this photo, well, can you get the right number? Of course you can. I mean, you may have to pick up a magnifier and look into the background of it, but you could estimate the number, but it will take you a while. But if I asked you, where is the man with the striped bathing suit? You will right away go to the right point. Why? Because we are using the stripes of the bathing suit as a salience concept to pull out information really quickly. If we think about the concept of salient apply into science, into data, we can see different ways of making something stand out. Grouping is the first one. You have something that separates from a main group and your eyes will absolutely land there really quickly. Size, orientation, shape, marks, motion, animations all over the place, and color. We will be talking a lot about grouping and about color just because we don't have much more time. But all of these can be applied into data visualization. So we're going to focus on the first part, on the grouping area. And I would like to quickly mention here the Gestalt principles. For this webinar, I actually did a stronger dive into the Gestalt principles than what I was trained for before, just to prepare myself. And I got really involved in it because there is a lot to learn about these principles. So the Gestalt movement was happening in the 1920s in Germany. And then it expanded throughout the world and it got updated over and over. So the original Gestalt principles are 6 of them. There are more added over time. But the idea is this whole concept rolls around the point of how we interpret what we see in a group, how our human eye processes visual information. And this can be applied to many areas. This can be applied to, of course, graphic design, but it can and it is applied daily to data visualization. So we talk about proximity. I'm going to mention 4 different Gestalt principles here that are important for grouping. So proximity is the first. We have 2 groups. And if I ask you, what are the 2 groups? You will say top and bottom because they are closer together, even though they are different shapes. The next one is similarity. If we keep the same distance between the 4 different shapes or the 2 different shapes, you will actually see 2 groups and you probably will tell me there are circles and there are squares. The next one is connection, which is stronger than the 2 previous one. If we connect the shapes with a line, the 2 groups completely break as they were before in similarity. And again, we have a left and right group. But even stronger than these 3 different principles is enclosure. If I use some kind of, and it has to be something really simple, you don't need to, overuse color or anything, but some simple shape in the background that will break anything else that you have created. And it will emphasize that the group now is top and bottom again. Now, how do we apply this to data visualization? Example of proximity, this happens all over the place. In many cases, people creating bar graphs, overmade lines, vertical lines of the y-axis to just separate groups. Things could be even more simple if you just use the proximity and the white space to separate the different groups. Similarity, again, it has been applied all over as well. We have different scatter plots that have, in this case, 2 groups, the filled dots and the white dots. And because they are similar, you link them together. This get broken by, or it can get broken by line, is the base of the line graphs. So connection really matters. And that's how we make groups that are connected by the same line. And then we have enclosure. And this is more powerful than anything else. And that's why it really is effective for data visualization. And you should keep it in mind when you want to highlight something very specific. Okay, that said, we're going to move into color because there is a lot to say about it. First of all, this is probably very basic, but it's always good to review what the color dimensions are. When we talk about color, color is a really general word. So we're talking, most of us think about hue right away. we have different colors, orange, green, blue, but also within color dimensions, we have to differentiate what is called saturation or chroma, which is the range between the full color and a grayscale. And then the last component, the last dimension of color, which is my favorite, I think that is the most useful one in data visualization and in scientific illustration. It has different names: value, luminance, lightness, or tonal range. Basically is how everything looks in black and white. That said, we couldn't skip the concept of color without talking about my favorite color system, which is the Mansell system. My dear mentor, Evelyn Mansell, introduced me to this beautiful Mansell color system years ago, and the more I read about it over the years, the more I get fascinated by it. Basically, Mansell was a German painter, an American painter of the 1900s. And so he was obsessed with understanding color and classifying how color works. And it's really fun for a scientist to understand how actually color is not arbitrary. There are many ways to organize it, but this system uses top and bottom of this scale to organize things between white and black, meaning value. It goes all the way around in circles to see how chroma or hue works. And then from the center to the outside, it analyzes how saturation or chroma is. The cool thing about this is if we represent this in 3D, we get something that is not a perfect diamond shape, which is what you could probably imagine up front. So not every hue has the same range, values change throughout the different hues, and the expansion that we can get in terms of saturation is different. This, I just point out here this cross-section of one of these parts of this three-dimensional representation that was published by Mansell, and you can see how it's not symmetrical. So that's quite interesting, just already understanding that we don't have the capability in our human eye to process colors equally. This is something good to remember when we make color decisions. Okay, stop talking about theory and color theory. We're gonna do an exercise. If I show you these shapes, we're gonna move now into color and link color to data. So if I show you these shapes, and then I show you these other shapes on the right side, and I ask you what has been happening here. Well, you probably guessed that we have organized shapes in groups in two different types of groups, right? So we have the first section that has used identity. We have triangles and circles and a diamond shape and a square. And in the second section, we have magnitude. That means something from a small to a large scale. We're going to use this classification to understand color. So keep this in mind, keep these two categories in mind, because we will be using them throughout the presentation. If we talk about identity, we're going to talk about categorical data. Data that can be put in different boxes, in different categories, and all of them are equally important. If we talk about magnitude, we are going to be thinking about data that can be ordered from a small to a larger scale, for example. That said, why I'm saying all of this, the color is coming. Here is the color coming. If we talk about identity or categorical data, we are going to use hue as the color dimension to play with this type of data. If we talk about magnitude or data that can be ordered, we're going to use the two other components, saturation and value. This can be a bit abstract to process, but you will understand it the more you use it and the more you see it. Let's mention something else about color. If we keep these two groups that we just mentioned, categorical data and order, and we classify different ways to represent this data, not only color, but others. Here are some examples. On the left side, we have spatial regions, we have color, but we have motion or shape. On the right side, in order data, we have position, length, area, color, value, and saturation, and so on. So if we make, it has been published, if we make a classification of what is most effective, it sits on the top of the page. We can see how a spatial region is the most effective way to show categorical data, and position, the most effective way to show order data. Why I am bringing this? Because color is not on the first line, especially for order data, meaning you might not need to overuse color when you are doing some kind of data visualization. So always make your decisions based on a clear explanation. Do we need color and why? Examples of categorical data, remember identity, hue are on the page. This type of data use color to indicate membership in a group, to cue attention, to organize or unify different elements. Now, the limits between what works and what doesn't work are not very clear, but in some cases are really obvious. In the center of this page, we have an example. Color for pure decoration is not indicated. It's not useful. It can bring a lot of confusion. If there are too many hues within a visual representation of data, we really cannot process this. So don't overuse it. Examples of ordered data. On the left side, we have a few. Basically, what is happening in data that you can order in a magnitude if you use color is that what we are using is a numerical scale that is linked to a change in color, to an evolution in color. Now, this is super useful and is used over the place a lot. But unfortunately, there are a very high use still of this type of color. It's called color ramp. This transition and this link between numbers and color is a color ramp. There's pretty much out there almost every day new publications still coming with a rainbow color ramp. And so I wanted to take the time to talk a little bit of why this might be a problem and how can we fix it. So many softwares are literally coming with this output directly. So you just create your figure, boom, done, and you have your rainbow color palette applied to the visualization. Let's use an example of two different areas within the rainbow color ramp. You can look at these two arrows, and we can point down the colors at the end of the head of each of the arrows. And you can see the problem already. We have a change from orange to this kind of lime, yellowish, greenish color on the left. And then on the right side, you have a change from blue to blue. And at least to me, on my computer, on my device, they look very similar. That means that for the same sections, that numerical section that we want to represent, the change in color, it really varies. So we can mislead a lot of the data that we have. Why? The mathematical explanation of this is really fun. There's not too much time. I will just mention it. But basically, if you move the color ramp of a rainbow ramp into black and white, this is what you get. You have areas that are very light and areas that are very dark in value. There's a fun publication from NASA that we can take a quick look that analyze the range that different hues have in value. And you can see how not all the colors are the same. As Mansell explained it already in the 1900 publications, the shape, how we can organize colors with all the hues is not equally distributed. That's why this type of ramp is actually not the best for data visualization. So for sure, rainbow, yes, let's leave it for the flags, but not for data visualization. And then somebody in the audience will say, well, there are exceptions always. I'm not setting in stone everything that I'm saying today. This is one of them. When we represent proteins and we talk about amino acid sequence, it has been very, very well established, this type of color transition. So we have to respect what has been established, and we can propose different alternatives for sure, but we have to know that there are always exceptions. But in general, try to stay away from a rainbow color ramp when possible. Keep talking about data, keep talking about color. I feel like always my take-home message is don't use color, which is not true, but just think about it, right? Don't overuse color. The overuse of color can create a lot of visual clutter, which is the opposite of what we are looking for when we create a visualization of data. Try to use color in a minimal way, emphasizing what you want to emphasize. It can really simplify complex tasks. An example of why using color without thinking too much about it can be a problem is this really wonderful example. You can look at and say, well, to me, it looks fine. It does. Until you think about how things look in a grayscale and am I being fair to the data? So in this case, if we translate this visually to a black and white approach, we can see how the light blue bands are totally underrepresented because they are lighter in value than the rest. That means that you are obscuring some of the data that you want to publish, and that probably is not the goal that you have. So when you think about color for any type of visual representation, make sure that you think about what do you need to express, what type of data you have, and how can you use color best. As I said before, color in isolation really works to highlight data. These are some of the examples where just a touch of color works much better than color all over the place, especially warm colors. Before I go into this example, a quick explanation. Warm colors are, we see this color palette, this color wheel, the one on the right side. Cool colors are the one on the left side. So if we use warm colors to put across any kind of visualization, data visualization, our human eye have a tendency to be attracted more for warm than cool colors, meaning, for example, in the bar graph that we have, you, even if you don't think about it too much, probably will get a perception of the increase over the x-axis versus the kind of equal distribution of all the bars, because warm colors are coming through your eyes first. Why we care about this? Well, this might be also important for making decisions. This is another example where we have the same graph that has been colorized with warm colors in two different ways. On the left side, we have colorized in red the control group, and on the right side, the patient group. If I show you the left graph only, you probably will look at the constant distribution of the control group. That might be not what we want to put across, right? So we might be able to probably put the focus on the patient data if we colorize data in a warm color. Something that I always love mentioning is that black and white are part of colors. They are also colors, and they work very well. And we avoid a lot of problems that happens with other type of colors. So, you know, we can use white backgrounds, gray backgrounds, or black backgrounds, and things can be colorized in black and white and work very, very well. There are different kind of feelings that we get depending on the type of background. We will talk about background a bit later, but the type of decisions that we make. But no matter what, there's always an option of black and white if you have that works very well. Now, I'm going to be giving you a few resources throughout this presentation. Don't worry about taking notes or whatever you need, because there's also at the end of the presentation a QR code that will link you to a PDF that you can download with all of these either interactive websites that are super useful, or resources, or references. So just keep listening. I wanted to mention one of my favorite, people ask always, okay, you are talking a lot about color. How do I make right decisions? How do I choose a proper color palette if I don't want to just come up with one on my own? And this resource is fantastic, the Color Brewer. It uses the type of classification that I have already explained for the different type of data, categorical or ordered data, to create color palettes that we know that are solid and work. The only thing is, of course, to make it a bit more fun, the names are a bit different, but it doesn't matter once you understand it. So this is just a screenshot of the website. Cynthia Brewer is a cartographer. This website is created to create maps and roads, and so the application of this resource on data visualization is really well established. But it's funny that the origin wasn't intended to be for everyone, just for roads. However, it works really well. If we look at the website, you can spend some time playing with it, but you can have your number of data series that you have input on the top. And then just something to remember, if we follow along this presentation, is that talking about categorical data and hue, it's called qualitative type of color palette versus the order data. The one that talk about saturation and value is called sequential or divergent, depending on the type of data that you have. Feel free to go on it and play a bit with it. It's really fun because you can download the color palettes directly, put them into Illustrator, Photoshop, or just get the numbers and input them in any kind of color system. So it works pretty well. This is just a visual summary of what type of palettes you can get from it. And of course, if we are talking about color, we cannot leave out accessibility and the concept of color blindness, and one of many resources that we could use to understand if the color palettes that we have are a problem for accessibility. So let's first of all mention the red and green color blindness. I'm just using the rainbow color palette. No, I'm not obsessed with it, but just as an example, because you have many types of hue. If you apply a color blindness filter on it, you can see how we lose a lot of information. So basically, we get two sections within the rainbow color palette. If I use the type of color palette that I decided to play with for this presentation, and I apply a color blindness filter, this is what we get. You can see how, again, we are losing a lot of information. We, from having 5 different sections, 5 different groups on my qualitative type of color palette, move into 2 or 3 color sections, areas. The type of color palette, the type of color blindness that are most dominant are deuteranopia. It affects around 5% of the male population, but there is a total of almost 10% of the male population affected by this, with other type of color blindness like protanopia or tritanopia. So keeping this in mind, how do we test if our data visualization is effective and it can reach everyone? I don't get paid by Color Oracle, even though I keep talking about it for a while, because it's a very small software that you can download, put in your computer, open it whenever you need to check something. And I really like that you can see how things look with different color blindness filters, but also in black and white, which is very useful. Implications in visual communication of this. Again, another example of a normal vision versus a color blindness approach. You again can see how this can really masquerade data and create confusion. Unfortunately, this the use of red and blue combined is very spread. It's still a lot in use. But if we can avoid the single use of red and green together, it would be fantastic, because the problem when we apply a color blindness filter is that we lose all that information. You can see that in the bottom. If you have to use these colors, there is a way to fix the problem. You can make the red a bit darker, the green a bit lighter, play with the value of it, and then things will improve in terms of color blindness filters and accessibility. Even better, if you can use other colors that are not those, you will probably stay more safe and be able to reach with data visualization to everyone. Unfortunately, the most widespread use of this combination of red and green happens in immunofluorescent microscopy, and there is a huge life science field on it. So when fluorescent microscopy is published, most of the times we get the phalloidine in red, and then we always get a green dye. So this is a problem for sure for color blindness, because you already have the red and green, not best combination of colors. You can see how applying a color blindness filter things are. How do we improve this? Well, it can be as simple as changing one of the dyes, either the red or either the green dye, and then the problem is solved. We cannot leave the color section again. There's a lot to talk about color. That's why it's my favorite area. But we cannot leave this section without mentioning background, because it really matters. And it not only matters for the data itself, but for the labels that we put on the data. So if we transform the hue that we have on the left to a black and white scale, we will understand why in the boxes that we have here, there are some problems. Why we cannot read very well, for example, the first combination of red and green. Well, if there's color blindness in between, for sure that's a problem. But also the hue of red and green that has been selected is really, really close if you move things to black and white. That's why having this filter of black and white is very, very useful. The goal that you will always be looking for or aiming to is to have a very high contrast in value between background and foreground. And here is a quick example. We have a visualization of a EGF receptor here, a very simple schematic. The contrast of the labels is not optimized. It's not the best. Can we improve it? Yes, we can. making the colors a bit lighter, so then we will be able to read it better. But can we improve this even more? Yes, again, moving to black and white, because black and white are always options, and not always, but most of the times, and they work very, very well. So don't overuse color. All that said, I need to mention the last resource. There's a lot of things out there to play with, but I love this. I love this website. This project was created to input online any kind of color palette and check how different type of data visualization could look like. You can turn on and off the black outlines, and you can check for different color blindness options and so on. So it's in the resource page that will be handy. Don't worry about taking notes, but it's really useful to just have a quick sense of, okay, do I like it? There's always a little personal preference here, so it is a good way to check things quickly. Okay, so stay with me. We're gonna move into the next part of this webinar, which is a very big, broad overview on graphs. Before we get into a specific, let me show you this. This classification is really useful. We are gonna talk about graphical perfection and the rank based on visual methods. What do you mean? So if I show you this, it has been proved that position is one of the best ways to portray data. Next is length, of course, using all bar graphs, angle, absolutely, area, and color. And again, why I am sharing this? Because color is at the end of the graphical perception and not at the beginning, meaning maybe using position works better for data visualization than just playing with colors. So these affect the way how we represent data. How do we make these decisions? Let's look at some examples. This is a New York Times example, is from 2015. There are many more updated ones, fantastic animations. The data visualization team in the New York Times is really fun. But anyway, as a quick example, what do we have here? We have color. We don't need to get into details of what this means, but we can see how color has been used in the scale, in the legend, not in the legend, in the oh my goodness, okay, the word is out of my head now, but in the key. So color is one of the ways how we can represent data. And then angle, the way how the different lines are angled, means something. We are not getting into the interpretation of this, but we can see how these two features have been used for this type of data visualization. More examples. On the left side, we have area, for sure. The how big are the circles means something. The position where they are means something else, and the color. And this is really fun because it has this kind of circular cue for color. So that's just a fun example to look at. On the pie chart examples, we use angle and we use area. And so there is always a combination of qualities that we can use to represent data. And the question is, which one is best? And I wish I had an answer, but there is not an answer. The best is, thinking about possibilities and playing with it and see what works better. We can see what works, and also we can quickly see what is not working. If I show you these two different circles and I ask you how many times B is larger than A, you can give me an estimate number and you might be able to, get the right number, but it's not obvious. It's 14 times and it's not something clear. If I show you this other pie chart and I ask you, can you order these different sections from the larger to the smaller? Well, you can probably get that Z is the larger one and it seems like B might be the smaller one, but then things get a bit muddy between A and D. And so can you do it? Yes. Can you interpret the data? Absolutely. But the goal in data visualization is always to create things that are very easy to understand, very simplified, very straightforward. You want to make sure that the message is put across as fast as possible. So maybe if I use the data that I have in this pie chart, in a bar graph, and I asked you the same question, you can tell me absolutely yes. I can for sure tell you what is larger and what is smaller. So that might help you playing with different visualization options to make decisions on what is working and what is not working. Okay, so I could be talking about graphs forever. There is a lot of information, a lot of really fun new different graphs these days to play with. But going to the basics, I feel like it's very useful for me, and I hope for you. So we're going to cover a few points. We're going to talk about the plotting symbols in graphs, access, line versus bar graphs. And again, this is very obvious, but it might be useful for you to train your eyes. We're going to talk about labels and the importance of the white space. There's a lot still to talk about, but it's quick. So first of all, the plotting symbols. Choosing the right plotting symbols. This may be something, you know, a small decision to make when you are creating any kind of data visualization with plots, with like symbols. But this is a big deal. Symbols have an impact on figure legibility and on the clarity of it. So we have to make two distinctions here. First of all, we have to distinguish if we are talking about a single data series, meaning one type of or multiple ones, because the approach is going to be a bit different. So if we focus on the single data series, the goal is to minimize data occlusion that is caused by overlapping symbol. In this example, everything is super expanded, clear, easy to read. But there are many cases where we have, we have line graphs where the data overlap with each other in a single data series even. And in this case, in every case, the best option is an open white circle, an empty white circle. And why this is the best symbol that we can use? We could use something else, right? We could use squares, triangles. We could use any type of cross. We could use anything. But it's very fun to see how the intersection of a circle with another circle, it doesn't create an image of itself. So if you pay attention to all the overlapping that is happening in the screen between circles, you get this kind of, fun inverted eye shape if you overlap two circles, but there is no the creation of a third one, right? However, with the squares, with triangles, with anything else, if you overlap it, if you have data that overlap each other, you may create some confusion because you create an image of itself. That's why if you have a single data series, the most useful way to represent the data points are white circles. Now, we talk about the single data series example, but what about the multiple cases? I cannot give you a very fixed rule about this, but the goal is to choose something that create strong visual boundaries. And what are visual boundaries? Basically, if you see these examples in the screen, you can see how comparing different shapes is very easy, right? Things are very different to each other. This is the goal, to visually differentiate the groups quickly. In the bottom, we have examples of symbols that have weak visual boundaries. So, if we just want to use triangles and depending on the orientation of the triangle, we can separate different groups. Can we do it? Of course we can, but it will take us longer. So as I said at the very beginning, simplify, makes things for the reader as simple as possible. Access, there's a lot to talk about access. And here are two examples of things that we can find out there. On the left side, we have a graph without access. On the right side, we have a graph with many axes. Both cases are used. It's very common. It's not very normal to have a graph without axes, but if you see one, stay away from it. Put your axes back. We need axes. Axes are the starting point of telling a story. So we need them. So don't get rid of them with no reason. Of course, there are exceptions. But also, there are a lot of softwares these days that are creating as an output graphs with the duplication of the Y and the X axis. And there is nothing wrong about it. But you normally don't publish with a single graph, right? You have a page with many of them. So again, the more we can simplify visually what we are creating, the better, because we are trying to stay away from visual clutter. This touches another of the Gestalt principles that talks about how visually our brain interpreted shapes and is able to add the missing pieces. So if we have this, And if we have the next one, if we have an axis that is only an X and a Y axis, visually we are putting the full box around, even though we don't have it there. So we don't need it because it's one of our mental qualities. It's something that the human brain can do. So if we can simplify again, let's simplify. Something that is more common than what we will expect is to see axes that are not properly scaled. So of course, make sure that you scale your axes well to show data properly. Again, part of the simplification and part of creating things simple. Every time that we can unify an axis, let's do it. This reads much better, is more simple, and you know, we can always use the break on data if we need to make the access common for everything. Another way to make things a bit more simple is to use shift axis. When at the origin, at the 0 point, we have data that overlaps. It's really useful to separate a little bit the 0 point, crossing point of the axes, so we can read the data better. Both, of course, the question, which one is correct? Of course, both are correct. Avoid unnecessary tick marks. And this is something that you look at and you say, well, this is obvious, but there is always publications when you look at the tick marks of the axes, and you feel like there is a lot of information that could be simplified, meaning it could be read quickly. We're going to do these very short exercises on bar graphs and line graphs. And the idea is to analyze the structure, and the idea is to train your eye to make things more simple. So we have this graph on the left side. It's quite, you know, it's from 2005, but still there are publications like that nowadays happening. And this is the makeover. So it's the same thing, but a bit better. What have we improved? We have get rid of patterns. It doesn't happen that much. It used to happen a lot that in some particular softwares, I remember doing my PhD, I used Origin. It had all type of fun patterns that you can use as an output in your graphs. And, you know, of course, people love it. But it doesn't work. It creates a lot of visual clutter, confusion. To avoid patterns, try to make things simple, try to use solid colors. And this is an example that I used at the beginning of the webinar when I talk about grouping and one of the Gestalt principles. Using grouping, avoid having extra lines, and this is the case. There is also improvement that we can do on labeling on axes, right? So the same exercise for a line graph, and I'm aware there are many other type of graphs out there for sure, but these two basic ones, you can apply the same principles to many other type of visualizations. Let's do a makeover and improve things. The main clear point to improve here is the type of symbols for a multiple data series. Simplify the plotting symbols is good. Making them large is good, so we can actually read it. And of course, if you pay attention to the y-axis, it has also been simplified here, which helps understanding things, again, faster. That said, a quality check that everyone should always do when you create graphs is to make them smaller. If you make things smaller, and the reason is, in most of the cases for publications, any kind of visualization is getting more and more tiny. So if you get things small and you can still read them, that means you are doing an okay job. And then just to finish, a last reminder, and there is not too much time, but to talk about visual completion, alignment, and the importance of the use of white space. There's a lot here, but just remember, sometimes when we try to create a composition of figures in a page, we just start from one corner and end up in the other, and that's it. It might be useful to pre-plan what we are portraying, because as I said before, our human brain have the capability of finishing or closing lines when we kind of see that some pattern is happening. So these are 3 examples where in yellow, there are some kind of background lines that will be removed in the final visualization, but are very useful to create things that work visually very well, and to help with how we read things and the order and the classification. That said, I know that we don't have too much time. Anna, feel free to stop me if we want to stop for questions. I just have some examples for practicing, and if we don't have too much time, the goal that what I want to put across here is go online, check publications, check data visualization, and think about how could I make things better? This is I want to be very clear that this is not a critique of the work that I'm just showcasing. This is just a way for me to train my eye and to show you how over time you can get better on this. Different publications, we're not going to mention, I mean, the references are here. It doesn't matter. There's nothing wrong about this. All of those are wonderful publications, but can we make things better? Yes. An example here, the problem with color blindness. We already, instead of four colors, we can see 2, which it might be a problem for the person interpreting this data. The axes are a bit small. The boxes is completely closed. We don't need to have the double encoding of the X and Y axis. Just some examples. This happens still a lot, and we tend to make things as fancy as possible, right? So we tend to make things as wild and crazy, and within this we end up adding dimensions to data that doesn't have that dimension. So this is a bar graph, a simple bar graph that has been just created in 3D. So we are adding a Z dimension when there is no dimension in that data. Besides, of course, rainbow and all of that we have discussed. So we don't need to add, make sure that you use the dimension of data that you have in your data set. Another example here, the box is closed, but there's a lot of problems with color in this example because we might be starting making groups visually. Look at the purples and we look at the dark reds, and we might be linking things that are not necessary to be linked, probably. Will this work in black and white? For sure. We probably don't need color here. Many examples out there that are really fun to understand and see how people solve. This is one of them where there's a lot going on in this graph. There's a lot of colors, and it might be a bit difficult to understand what they mean. We might be linking things that we shouldn't be linking visually. Can we make things more simple? Yes. Use color to highlight data, use warm color to highlight what you want. More examples, gigantic data plot, gigantic key points that are really masquerading, obscuring data are not necessary. Vertical lines can make things maybe a bit too complicated. And really important thing that it's, sometimes totally overviewed is tick marks facing inwards, facing the space of the data visualization. Try to avoid that. Try to put tick marks looking outwards so they don't interfere with the data. More examples, experimental data that links visually colors of what is happening in these skin areas that we want to emphasize or we want to highlight within the colors of the graph. We try to link the red with the red, but actually they have nothing to do with each other, so this might be a problem. Red and green. Again, color blindness, please keep in mind, don't use these color combinations. Not everything is bad. I'm just choosing things as I go, but this is this is one of the examples of using color in a really small amount and highlighting data with warm colors. The example on the right side, a lot of colors over the place. We cannot see what's going on. And then there is actually a yellow line that is really hard to read. Structural biology papers are wonderful and full of colors for the different areas of protein visualization. But then when you apply color blindness filters, we have to be careful because things can get a bit confusing. And I could keep talking and talking forever about examples, but I think we should leave a bit of time for Q&A. Before I go, there's a QR code on the page. We can send it out as well or put it in the chat if necessary, but please scan this QR code. It will take you to a PDF that you can download, and you can have all the resources that we have been covering, very, very important publications. Of course, just some examples, there is a lot out there, but I hope it's useful for you. And with that said, I think we are at the end of the webinar, at least the presentation. Thank you very much.
Wonderful. Thank you, Isabel. I think that's given us all lots of things to think about. We will make sure to share the link for your extra resources along with the recording when we send out the email for that. So we'll share that with that too. So just before we get to the questions, we have a little time for some, and I can see there's already some in the Q&A box there. But I just wanted to mention about upcoming webinars. So We're going to be taking our summer break now for the next couple of months from webinars, but we will be back in September. And in September, we're going to be running a series on spatial omics and single cell. So keep an eye out for that. Registration will be coming soon. So keep an eye on LinkedIn and wherever else you've seen our webinars mentioned. And yeah, we'll be back in September for that one. In the meantime, we have plenty of recorded webinars that are available. So all our webinars do get recorded and are made available in the on-demand section of the training EMBL-EBI training website. So do go and check those out. Okay, so I think at that point we can have a look at some questions. So I can see some coming through already. There's some that are quite popular, so let's go and have a look at those. The most popular, which is a question around what software do you use for figure preparation?
For figure preparation, well, keep in mind, I'm not in the lab anymore. I miss it. So I don't do that much of figure preparation itself coming from my own data. But I have done and I do quite a bit of improving visualization itself. So no matter what program you use to create the data, it's always useful to bring it into softwares like Illustrator, in my case, is what I use. I know there are alternatives to that, but places where you can guarantee that the data stays where it is. I can bring a plot that is a scatter plot into Illustrator and make sure that I'm not moving any dots of my scatter plot. But I can play with the thickness of the axis. I can play with the colors. I can play with the thickness of the different data sets. So there is the scale of the numbers in the axis matters a lot. And so Illustrator gives you a lot. In my case, I'm not promoting Illustrator. There are many other softwares for it for sure, but that's what I use. Once you have the data in whatever software that you want to use, depending on your area of work or expertise.
Great, thank you. The next question we have is around whether you have any books that you would recommend for further reading. Obviously, you've given some ideas in your resources list about publication.
Yeah, there are some books. Honestly, I wish I had more time these days to read books. But I end up falling asleep before I read anything. So I tend to go online a lot for a lot of online resources more than physical books itself. But there are wonderful books out there for sure. There are some of them listed on the PDF that I share. And if somebody has any particular area that wants to dive into, I'm happy to have a one-on-one chat for more particular recommendations.
Okay, thank you. We have, well, we can't have a webinar without having AI mentioned at the moment. So today's question is, how do you see your work changing with new AI tools being available?
I see efficiency and I see excitement. I know that I have to be cautious. We have to be cautious. So I see a lot of scientific check on output coming from AI and making sure that accuracy is respect and maintained. There is, I think that in general, in scientific illustration, which is my main area of work at the moment, I think that AI could be useful for brainstorming. It could be useful to look at other points of view. As a final piece, no, at least for scientific illustration, because there is no double check of scientific accuracy, right? So you end up with DNAs that are turning the wrong way, or hands of people that has 6 fingers, or it's there's no check control, there's no quality control, and the same thing happened from any kind of data visualization approach or creative writing happening with AI. We can for sure look at it, we can play with it. I am for sure totally up for playing with new tools. But overall, we have to keep scientific accuracy. I think that should be never misled or lost. So that's priority.
Okay, thank you. I think we have time for a couple more questions. So you shared a few images there that had error bars on. Is there an alternative or better way to represent that kind of information?
That's a really good question. For error bar, well, general thoughts, as I don't have a section on error bar particularly to think about this, but it's for sure something to elaborate more. It happens a lot that you look at data visualization with completely out of range error bars that are either too small, that are either too large. Some people, you know, in a bar graph, for example, use half of the error bar that has been okay in the field. In some other fields, that's just not cool. So I think there's a lot to do depending on your particular case. There are some examples, for example, of or area representations of different areas with a lot of error bar that masquerade the data itself if you over put lines, the thickness of your line of the error bar. So I think there's no one particular thought on it. I think depending on the type of data, just use common sense. Is the error bar of this graph overcasting the data? I may have to play with the color of it, or I may have to look at, you know, other thickness of lines, or is my error bar completely misleading? So I think that I don't have a particular answer for it, but using common sense and thinking about the principles that we have discussed.
Great, thank you. Let's go for our last question. Someone asks, I've seen some comments suggesting that circle graphs such as pie charts are less effective or even problematic in certain contexts. Do you think that's true? Do you have a sort of an opinion on that?
No, I don't think there is any problem with circle bars or a pie chart. In fact, I use one for my presentation all throughout the slides because I really like them. We just have to know the limitations that they have, right? So we have to, again, understand what can they be used for. Do we want to use angle and area to put across some information? Well, as I showed before in one of the examples, it might not be the best way to visualize something. For example, if we have a qualitative type of data with 200, with a data set of 200, we might not want to see a pie chart with the 200 separations on it. If we have 3 groups, it might be very effective and very clear. So again, I wouldn't say they are not good. I don't think that there's nothing wrong with anything, except that we need to use common sense and we need to understand the data. And we might use a pie chart and look at the data and feel like, well, maybe I thought this was the best, but maybe I need to switch because the data is getting a bit difficult to read. At the end of the day, there are many ways to explain something, but what we are looking for is to explain it quickly because we want to explain a lot most of the times.
Great, thank you. Okay, so we'll leave the questions there for today. We're coming up for just an hour now. So I just wanted to, before we finish, mention again that we will share the link to the resources along with the recording tomorrow. You'll receive an email, so don't worry if you missed that. You will get that one. And in there was a couple of the different resources that have been coming up throughout the slides. Just wanted to, at this point, say thank you to Isabel for coming along and joining us and sharing such an informative and interesting talk today. And thank you to everyone who joined us for today's webinar. You will see again a little survey pop up at the end. So we really would appreciate your feedback if you're willing to do so. So we hope to see you again at a future webinar. Thank you.
Thank you very much, everyone.